Good evening, everybody. On behalf of the Department of Botany and IQAC of Gurudas College, we welcome all participants to the 13th webinar organized in our college, Gurudas College. I would just like to introduce all those present in the digital studio. We have Dr. Moushami Dash, Associate Professor, Department of Botany. We have Dr. Joydeep Kondo, the librarian of Gurudas College. And we have a resource person, Dr. Anirban Kar, all the way from USA to deliver his talk. And I think we'll be joined shortly by Professor Tripurna Majumdar, IQAC convener in our studio, and myself. I am Dr. Neetu Dev, the present head of the Department of Botany. I think Professor Tripurna Majumdar has joined us. So, Tripurna, welcome to the digital studio. I was just introducing all our participants. We'll go straight away to our program. So, Joydeep, could you share the message from the principal, which I'll read out, due to some, as the principal is very busy, she could not be present physically in the digital studio. So I'll just read out her address. So Joydeep, please share the slide, please. Okay, I'll read the message from the principal. It is indeed a great pleasure for me to announce that the resource person of this webinar, Dr. Anirban Kar, is an alumnus of the Department of Botany, Gurudas College. Today's webinar on understanding the complexity of cancer progression and biology of DNA structures would be interesting to students of life sciences as it is a part of the syllabus of many life science subjects, both in the undergraduate and postgraduate level. I hope that participants, especially students, are enriched listening to Dr. Anirban Kaur, who has many years of research experience in this field. Dr. Moshmi Chatterjee, Principal, Gurudas College. So now, after the principal's address, may I ask the IQAC convener, Professor Tripona Mojumdar, to say a few words.
Thank you, Mitu. Good evening, everyone. Hope all of you are keeping well in these trying times. On behalf of the Internal Quality Assurance Cell, Gurudas College, I welcome you to today's webinar. This webinar is the 13th one organized by us. Today's webinar is jointly organized by the Department of Botany and Internal Quality Assurance Cell of our college. Our speaker today is Dr. Anirban Kar. It is a well-known fact that the quality of an educational institute is often linked to the quality of its alumni. Anirban has graduated from our college from the Department of Botany in the year 2004. He has really made us proud with his extensive research work in laboratories in India, reputed laboratories in India, as well as in the US, and several publications in journals with high impact factor. My best wishes are with him. Today, he will speak on understanding the complexity of cancer progression and biology of DNA structure. Cancer is a dreaded disease. Knowledge about it for people like us will help a lot, since we do not have any have a positive attitude towards this dreadful disease. Moreover, I hear that this topic is a part of both the undergraduate as well as the postgraduate syllabus of our university. Hence, it will help many students of life sciences in Calcutta or elsewhere. Without wasting any more time, I request Neetu to continue with the proceedings. Stay safe, stay healthy. Thank you.
Thank you, Triponadi, for the introductory remarks. Now I call upon Dr. Joydeep Chando, the librarian of Gurudas College, to say a few words on the Moodle, which powers all these webinars and all in our college.
Thank you, Mitudi. It's a great pleasure for myself and for library to say something these esteemed participants. On to our Moodle. Moodle is a learning management system. And to describe it, I just want to share my screen for 2 or 3 minutes just. So I think I am audible, Mitudi. So there is a module system. Actually, it is Gurudas College Courseware. Actually, it is Gurudas College Courseware. To go to it, you have to first log in it. You see that there is a login site actually, and when you log in it, you just see your name, say for Mitudi, Mitu or something, etc. And this is our home page. You see the beauty of the site. It has been visited by more than 23,298 times. And it is done actually the visit, most of the visit occur during the last 2 or 3 months. And actually we choose the system, we have chosen the system during the preparation of next SSR. And last year when you just go through the next SSR, we see that there is a MOOCs, that is massive online open courses, and they have recommended to use the learning management system. And for this we have created this particular system. You just see that not only that, we have incorporated the total system. Total system means the total e-content of our faculty members. Faculty members can take online class through this particular system. It is the online class of botany. You go to the inside the any courses, you see that different online classes, different materials, e-contents of especially faculty members of different departments. Here you see Mitudi, Momita, then Mona Lisa, then Moushumidi. All e-contents are given there. Okay, so that's the beauty, and student can easily download. Just see that, easily download any material from this particular module database. Not only Word file, not only PDF file, here we can incorporate or embed different YouTube file also, or audio file also. And not only that, we have incorporated here all the webinars of the colleges. And you see that today is the 13th webinar. The details of webinars, the YouTube embeddations, all things are given there. You see, just for example, Gurudas College webinar, Coping with Stress and Managing Education, which was held in 13th July 2020. And you see that there are YouTube links. If you want to open the link or click on it, the link is open and the video is going on. That's the beauty of the total system. And not only that, just that, see that it is starting now. Okay. And if you want to see the details, feedback forms or analysis, you can also see here. Just go to feedback form. You can analyze the answer here. So what is your present status? Different questions are there, and you can analyze within a very few minutes about every webinars. So the beauty of the module database is that you can organize webinar within this. You can organize different online class within this. You can organize different assignments or different examination within this. You see that UGC Net Coaching, it is now prepared for organizing different online classes or online examination also. Okay, so through this webinar, we are requesting students, faculty members from different institutions, please go through this so that you can get different reading materials, different webinars, different video materials from the faculty members of our college. And after you're using this, you can also provide us suggestions, and we are very much grateful to Bengal Library Association. They have totally customized this particular module system. So again and again, on behalf of Gurudas College Library, I thank you all for giving me the opportunity. Thank you, madam.
For this description of the Moodle. Many people are not really aware of the whole system. So good of you to show it in your presentation. Now, I'd like to call upon Dr. Moushumi Das, Associate Professor, Department of Botany, to introduce our speaker.
Thank you, Mitu. Good evening, everybody. It gives me immense pleasure to introduce Dr. Anirban Kar as today's speaker. And I'm also very happy to share with all of you that our speaker is our alumnus. Dr. Kaur did his graduation from Gurudas College in Botany and MSc from Bhopal University in Biochemistry. Subsequently, he did his PhD at CSIR IGIB under supervision of the eminent scientist Dr. Shantanu Choudhury, our Shanti Swarup Bhatnagar awardee. He worked extensively on DNA secondary structures and cancer metastasis. And in the process, he developed a keen interest in telomere biology. He and his mentor jointly received the prestigious DBT India Wellcome Trust grant to work on the telomere biology in India for the first time. After completion of PhD, he moved to USA to do his postdoctoral training with world-renowned electron microscopy expert and telomere biologist, Professor Jack Griffith at the University of North Carolina at Chapel Hill. At this young age, he has achieved a lot of academic excellences. I'm just mentioning some of his major career milestones. He has quite a number of good publications to his credit in peer-reviewed international journals and book chapters. He is also the member of reviewer panel for several international journals, including Nucleic Acid Research. Dr. Kaur today will speak on understanding the complexity of cancer progression and biology of DNA structure. I wish Dr. Anirban Kar all success in his future endeavors. Thank you. Over to Dr. Anirban Kar.
Please adjust your camera. Dr. Anirban, please adjust your camera.
Yes, I was trying to, hi. I was trying to unmute myself. So, okay. Good evening, everyone. আমার মনে হয় এখন ওখানে সন্ধ্যাবেলা, সুতরাং good evening. I would like to thank everyone, especially Department of Botany, Riporna ma'am, Joydeep sir, for organizing all this. So when I left my college, I moved to do my master's. I never thought I would do PhD. But after doing my PhD, I still wanted to do more research. So I moved to US and I continued my PhD work here with one of the greatest electron microscopist and field expert in telomere biology, Dr. Jack Griffith. Then once I finished that, I was not done. So I moved into NIH to continue some more different works. But it's a long journey. It's almost been 14 years since I started this. So let's see. আমাকে বলা হয়েছে যে এটা আমাদের আমি দেখেছি যে এটা আমাদের সিলেবাসের একটা পার্ট। সো আজকে আমি বেসিক্যালি যেগুলো আলোচনা করব সেগুলো ক্যান্সার বায়োলজির বেসিক ক্যান্সার বায়োলজি। আই এম নট গোয়িং ইনটু অ্যাডভান্স ক্যান্সার বায়োলজি। আর নেক্সট half of the presentation will be mainly the cancer biology and basics of cancer biology. The other half will be telomere biology and DNA secondary structures. Basically, throughout my talk, I will try to show you that how cancer biology can be so basic, starting from very long DNA structures at the very end of the chromosomes, which is interesting because আমরা যখন ক্যান্সার দেখি আমরা ক্যান্সারটাকে একটা নর্মাল ডিজিজ বলে ভাবি এবং আমরা খুব কম লোকই আমরা জানি যে ক্যান্সার কতটা কমপ্লিকেটেড। সুতরাং আমার আজকের প্রেজেন্টেশনটা বেসিক্যালি আমি ট্রাই করবো যে দেখানোর জন্য যে ক্যান্সার as a disease is a very complicated disease. সুতরাং okay, let's not waste any more time. So to start with, today's title is Understanding the Complexity of the Cancer Progression and Biology of DNA Structures. Now, the talk structure today will be like half of the talk is dedicated to biology of cancer. I will discuss the causes of cancer briefly, then brief idea about carcinogenesis, how cancer actually happens, then a brief talk about the metastasis, how cancer progress or spread throughout the body, then a little introduction about cancer staging, so that once this talk is over, we know how, when we see a doctor's report, that how different stages of cancer are classified. Then concept of proto-oncogenes and tumor suppressor genes. This is a part of the curriculum. Then moving into the other part of the talk, it will be purely telomere biology and DNA structures, which is again part of your curriculum. And I will introduce you to telomeres, the telomere structure. Then telomeres is a fascinating enzyme, so probably you will love to hear about it. It's one of a kind of enzyme in a human body. There is none other like this one. Then I will talk about telomeric structures. So it's basically a telomeric loop, or we call it T-loop. And then we will talk about G quadruplex biology. So this is what I did during my PhD. I still do it, but not much. And this is a fascinating thing to know that DNA, being a simple double helix, can go beyond double helix and form something which is much more fascinating than a simple double helix. So to start with, cancer. So cancer, when we talk about cancer, we generally think that cancer is a human disease, or maybe animal, plant sometimes. But the interesting part is cancer actually evolved throughout the evolution of Earth. So this, in the slide, you can see that on the right side, this bone, this is not a human bone or an normal animal bone. So recently, like 2 days back, I heard a report that actually paleontologists, they discovered that cancer was not only happened back during the dinosaur era, but they actually found this bone is actually deformed osteosarcoma bone belonging to a dinosaur, plant-eating dinosaur known as Centrosaurus. So cancer was there at that time, which is fascinating to know that, because when we imagine cancer today, we always think it's a human disease or maybe animal disease, but no, this was a dinosaur bone having osteosarcoma. And people actually looked into different dinosaurs, and they show that dinosaurs were having cancer like osteosarcoma, and they also used to have arthritis, which is interesting to know. So cancer actually evolved with the earth, and now it reaches the human, and so it's not only a human disease. So let me make that straight. Now, the features of cancer. So generally, the cancer, when we talk about cancer, it's a very broad term. It's not restricted to a particular kind of disease. It's an assembly of diseases. Cancer doesn't only infect a particular tissue. It can go in blood cells. It can go to lymph nodes, anywhere it wants. So it basically describes any kind of cellular changes with uncontrolled growth and division of cells. Now, the cancers can also basically result from abnormal proliferation of any of the different kinds of cells in body, and hence we have now know that there are more than 200 types of distinct cancers which we can classify. Now, some types of cancer, generally when we talk about cancer, we always think that cancer is basically rapidly cell growth and it will burst into a tumor. But no, cancer cells can also grow slow and stay for a very long time in our body and grow with a very slow rate. Now, there are cancers where you can see a tumor, which is generally what we visualize about cancer. But leukemia doesn't have a tumor. It doesn't form a tumor. Now, very recently, actually, long back, when cancer actually people started studying cancer, they also knew that there are various genetic factors which can contribute to the development of cancer. Now, looking at these two pictures, there's an interesting fact that you will find that all over the world, there are different percentage of occurrence of cancer. But if you look, two things. One is the American, which is 21% cancer incident. Now, look at the America's global cancer mortality rate, which is 14% only. So they have, although there are 21% people who are getting cancer, but only 14% are actually dying. So 7%, they can actually, 7% people are officially surviving. There can be different cases although. But on the other hand, in Asia, you can see that the percentage is huge, which is 48.5% approximately. But look at the mortality rate. It's 57%. So there are more people dying. So actually, this increases because there are post cases, which are like they have been treated, they have been known to be survived, then it comes back. So the mortality rate increases. So the objective of cancer biology or the work we do is to understand why that is the case or how we can treat this. But I will show you a slide where I will show you that why it is so complicated. Now, causes of cancer. In a small term, we can say cancer can be caused by almost everything in today's life. It can from lifestyle, which is like smoking, lack of exercise, or other unhealthy diets. It can be environmental factors like the pollution, other carcinogens. It can be pathogens, which I will show you in a slide, that viruses, although now we are in the COVID era, we know about viruses quite a lot, but viruses are actually cause basically almost 20% of cancer in the world, which is huge number. Then there are diseases like obesity, diabetes, immunosuppression, which can actually cause cancer. And the last, but one of the most important, is the genetic mutation. So all of this sometimes can cause genetic mutation in the body cells, and which can cause cancer. Now, right now, my work is basically based on this, genetic mutation, understanding how DNA damages can cause this mutation. So let's see. So DNA damage. So one of the prime cause of genetic mutation is when our body cannot repair DNA damages. So what are DNA damages? So we know about DNA double helix. So our DNA double helix every day gets bombarded with different kinds of assault, like oxygen radicals, which is produced inside our body cells. So it's internal. Then we have radiations, chemotherapeutics, UV light, and ionization radiation. And it is fascinating to know that each of these insult actually causes different types of DNA damages, completely different type. And that's what we study right now. And this, every assault or DNA damage can be repaired by particular mechanism, like base mismatches. We have single-stranded DNA breaks, so one strand will be broken. Then we have DNA adducts, where different chemical molecules can go inside the DNA and bind them. Then we have double-strand breaks, which is generally the major cause, one of the major cause of cancer outbreak. Now, each of them can be repaired with different mechanisms. For mismatches, we have mismatch repair. For single-strand DNA work, we have base excision repair. Then we have DNA adducts and nucleotide excision repair repairs it, followed by the double-strand DNA breaks can be repaired by double-strand DNA breaks repair, which is very simple. Okay. Now, my current work is basically focused on these three parts of DNA damage repair mechanism. Now, so what happens? So when we have a DNA damage, And you can see that the DNA damage is then repaired by different mechanisms, which are this, which I told you. Now, when we have the repair, the cell survives, fine. But when we don't have the repair here, we have different kinds of things happening inside the cell. If the cell is not happy, it will go into cell cycle arrest, or it can harbor some mutations, which is not the original sequence, some mutated sequence. We can have chromosomal instability, aging, disease, apoptosis, or cell death. But one of the things, which is actually an accumulation of all of them, is carcinogenesis. So, or cancer progression. So my current work is very complicated. I will give you a small, short glimpse what I do. So I told you that I work on 3 different cancer DNA repair pathways. So we have observed, and people around the world have seen, that these DNA pathways generally work to repair a kind of DNA. But what I am doing right now is basically to look into the broader or bigger picture. So if we look into proteins which actually do the job, because proteins generally do the function in the cell. So we have proteins from the base excision repair, which is BER. Then we have proteins from the double strand break repair, and we have proteins from other DNA repair pathways. We have seen that these different proteins can actually talk to each other and coordinate different kinds of DNA damage repair. And not only that, they can interfere with each other's job. So each of these blue lines actually is I would say, these are speakers. They are talking to each other. This is their way of talking. So this protein from here can talk to a protein from other network, or it can talk to another protein which is talking to the another network. So this way we are building a network of DNA damage repair proteins to understand how, when one of these protein is overexpressed, like not in its basic expression level, or its expression level changes, maybe over or under expression, it can actually form a network which actually interfere with the other network, causing possibly cancer, or probably enhancing the tumorigenesis process. Now, I told you that I will talk about oncogenic viruses. So as I said earlier, that there are almost 15 to 20% of total human cancers are caused by viruses. Now, generally there are many more viruses causing cancer in other animals, but only 7 of them till now has been recognized for causing cancer in humans. So these are very famous, those famous 7 viruses. We have EBV, it causes Burkitt's lymphoma, Hodgkin lymphoma. It's basically any kind of lymphomas. And then we have KSHV, which is Kaposi sarcoma herpes virus. It causes primary effusion lymphoma, Kaposi sarcoma. Then we have different kind of high-risk HPVs. Then we have MCPVs and others. So cancer is not only caused by the environment, it can cause by the viruses too. So sometimes it's not in our hand, because if we get infected with these viruses, it can lead in the long term to carcinogenesis process. Now, let's talk about hallmarks of cancer. So when I was doing my PhD, there was a nice paper from 2 prime people in the world of cancer biology. One was Douglas Hanahan and another was Robert Weinberg. They wrote an excellent review talking about what are the characters of cancer. So this was 2000, and in 2000, they actually identified 6 different hallmarks of cancer. One was the sustaining of proliferative signaling. So the cells will continuously grow. They will not stop growing, which probably all of us know. Then we have evading growth suppressor. So to grow, you need to stop those factors or proteins which stop growth. You will stop expression of those proteins. Then we have activating invasion and metastasis. So the cancer cells need to move out. So they activate a program through which they can go out and infect the different parts of body. And then we have enabling replicative immortality. So the cells live forever. That's one of the features probably we all know. The cancer cells doesn't stop growing. They live forever. And then we have angiogenesis. So it's one of the complicated part of cancer biology, that the cancer tumor can do angiogenesis. So it can actually make new blood vessels because the blood vessels carry food, oxygen to them. So they create new blood vessels. And then resisting cell death. So these two, immortality comes from resisting cell death. But inside the cell, they are two different biology. So they are actually trying to stop cell death. Now, but this was not it. So after 11 years, these two people, Hanahan and Weinberg, came back because they figured out cancer is not these 6 hallmarks. They are basically much more than those 6. So they have identified now 11 or 10 hallmarks constitutes the basic cancer network or cancer development. So other than these 6, now they have genomic instability, which I work on right now. I worked on this activating invasion of metastasis during my PhD, and I worked on this part during my PhD and my first postdoc. But they also showed that cancer cells actually deregulate cellular energetics. So basically, what they do is they deregulate the whole energy biology of the cell. So cancer cells basically prefer glycolysis. So they can survive very energy deficient conditions. Now, there are other things like avoiding immune destruction. So immune cells generally pick up which are the bad cells in our body, but cancer cells can escape immune system. They also cause tumor promoting inflammation. So this all together becomes a new hallmarks, or what they called it hallmarks of cancer next generation. So probably down the 10 years, we'll have another new hallmark system because cancer is always evolving. That's one of the worst part of cancer, that it evolves. It always adapts. That is very important, biology of a cancer cell. Now, I told you that the therapeutic targeting. So যখন আমরা basically normal লোকেদের সঙ্গে কথা বলি, আমাদের বাবা মা আত্মীয় স্বজন, ইভেন আমিও, অনেকে আমাকে জিজ্ঞেস করে যে ক্যান্সার ট্রিটমেন্ট কবে আসবে। So let's see, cancer treatment কবে আসবে। So cancer biologists all over the world actually are working on all this together. So they are trying to stop one of those hallmarks each at a single time. So suppose to sustaining proliferative signaling, we use EGFR inhibitors, or we used telomerase inhibitors to stop enabling replicative immortality. Now, we also used PARP inhibitors for genome instability and mutations. But the problem is everybody is trying to stop one hallmark. There is nothing which can stop all of this together. But a cancer cell is basically all of this together. That is the problem of cancer therapeutics today. So you need a mechanism or a treatment which can answer at least quite a few of them in a single go. And they are very connected. They are not separated from each other because a single cell, they have all this feature. So you need to escape each of them in a single treatment. So that is the complicacy of cancer therapeutics today. But probably with our human inventions and everything, we will get there someday, but not very soon. Now, this is the biology of a cancer cell. So how a cancer cell is generated. So first, the cells is genetically altered, which is probably epithelial cells. The alteration can be caused by mutations, multiple mutations, or something else. Then it goes into hyperplasia, which is basically cell divides more rapidly than normal. So it just keeps growing. Then once it reaches a size or mass, it starts changing its form. A cancer tumor, if you see a cancer tumor and cut it half, a cross section through it, you will see it's not a one type of cell. Probably it's been originated in your skin. But once you take out the tumor and try to see through it, you will see it's not a single tumor. It's not a single cell. There are multiple kinds of cells which are there. So this cause is termed as dysplasia. Then it starts the in situ cancer, so a tumor, which probably you can visualize. But then the worst thing happens. Once that tumor reaches its critical mass, which is debatable, but it started to come out of the tumor, those cells, and then these cells go into the bloodstream or lymph nodes, and then it starts moving into distal parts of your body. Now, I will talk about metastasis shortly, but let's see. Then it starts moving out and it causes metastasis. And hence, you have all the issues all throughout your body. Now, the process of carcinogenesis or oncogenesis. A lot of people generally think that carcinogenesis and tumorigenesis are the same. No. Tumorigenesis is formation of a tumor. It can be a benign tumor. Just sitting on your body, doing nothing. It's a mass. You just cut it off. It's good. But when that tumor has a cancerous property, then it's a cancer. So carcinogenesis is basically the development of cancer, or oncogenesis is the same. So the process of carcinogenesis leads by the changes like cellular changes, change in cellular phenotype, then genetic changes. The genetic structure of the cell will be changed. The cancer cells, which I will talk very soon about, they will change their genetics. They will activate some genes, they will deactivate some genes. Then it can go beyond genetics, which is, we called it epigenetic. So now the cancer cells can actually activate some genes which has been silenced probably since your childhood. Then it goes into abnormal cell division. So it's a multi-step process. It's not a very single one-shot go, like it will happen and you will become a cancer patient. No, it's a very long and strenuous process for the cell too. And also people have shown all over the years that cancer is not caused by a lot of cells together. It's basically a very small fraction of cells which form the cancer. And we call it tumor initiating cells. Or in modern cancer biology, a lot of people like to call it cancer stem cells or CSCs. So they have stem cell-like properties, which actually the stem cells, probably a lot of us know that a stem cell can actually try to replenish a different part of our dead cells and different parts of our bodies. There are adult stem cells, there are embryonic stem cells. So these are like stem cells. They can grow and regenerate themselves. Then these tumorigenic cells, the cancer stem cells, are believed to be the driving force of tumor progression. So this small amount of cells go out and create this havoc. Now, I basically made this slide for the students so that you can have a clear idea how we differentiate in terms of cancer biology, the different kinds of cells. It's a busy slide, so I will just summarize it. So there are different kinds of features which makes a cancer cell not different than a normal cell. The growth factor proteins. So the growth factor proteins are basically different kinds of proteins which promote growth. Now, in normal cells, generally they stop making this once they have reached their growing out of their growing phase and they don't need it, but cancer cells continuously produce this for their sustenance. Now, you have specialization and maturity. So normally a cell is destined to be a particular type of cell. So like I would say an epithelial cell, a skin cell, is probably different than a cells lower than that. But the cancer cells don't like that. They like to be in a condition which is non-differentiating condition. So whenever they need, they can differentiate into something different. Then we have cell-to-cell interaction, which is one of the hallmark of cancer, is that cells, when they come into contact with the other cells, they stop growing over them. But cancer cells don't do that. They grow over them. And that's how they form the tumor mass. Then they can escape apoptosis or the programmed cell death. Their morphology can generally, for normal cells, it's uniform, but cancer cells don't have really a morphology, kind of particular type of morphology. They can change. And also their nucleus can look darker under microscope because of the increased DNA strands. Then we have response to immune system, angiogenesis, which I told you that generally our body create new blood vessels to repair the damaged tissue, but cancer cells can do that same thing within the tumor. Then we have chromosome quantity. Generally in normal cells it's defined, but in cancer cells it's not defined. It can be more than 46. Then we have telomeres. I will talk about telomeres soon, so I will not go into, but just for knowledge that when a cell divides, the telomere gets shortened, but in the cancer cells, they have a mechanism to make that thing going on and on and on so that they can live. So telomeres are one of the reason or one of the way which we call a way towards immortality. So they can live forever, actually live forever. So let's see. This is the one of the picture I like about cancer metastasis because it shows how complicated cancer metastasis is, or how painful it's for a cancer cell even to do metastasis. Now, this is your primary tumor. So you can see there are different kinds of things in the primary tumor. You have T cell, you have tumor cell, original tumor cell, you have blood vessels, and then you have cancer-associated fibroblast, which helps to maintain the physiology. And then a lot of different things. So what happens is that the cell, the tumor cell from here, break the tumor mass, invade the lower tissue, which is we call invasion. Then it goes into blood vessel, we call intravasation, so moving inside the blood vessel. Then it goes into circulation. So he is now moving towards different parts of body. Then they go out of that blood vessel. We call it extravasation. Now, all these things are very tough thing to do for a cell. It's very hard for them to live through all this torture, but they still live. Then they reach a particular organ, suppose lungs, and suppose this originates from breast, it will move to lungs and from a pre-metastatic niche. Now, pre-metastatic niche is basically where they just enter the system of that tissue or organ and then form a small micrometastasis. You probably cannot even visualize this. It's very hard to look. Then they sit there. That's the most important part. They sit in that dormancy phase, which is this one, for a very long time. It's years, sometimes months, sometimes years. And they just sit there, doing nothing. Then suppose somehow your condition of the body deteriorated due to some chemotherapy or maybe something else. What happens? That triggers these cells. And they then becomes another tumor in a different organ. So this is how they move, which is very tough. Now let's see some features of metastasis. As I told you earlier, that it's a general term we use to tell people that the spread of cancer from primary tumor to different tissues or even to a different organ. The interesting part is metastasis causes 90% of cancer death, not the primary tumor. So this is the most important part of, or deadliest part of metastasis. These are the 5 steps which I told you earlier. Local infiltration, then transendothelial migration or intravasation, survive through the circulatory system, extravasation, and subsequent proliferation in competent organs. Now, metastasis is also very inefficient process, as I told you. Only 0.1% of the whole disseminated cancer cells actually form metastasis, but that's enough. So that because the process generally kills maximum of the cells, they don't survive the process. Now, metastasis progression can be early or late event. So there is a hypothesis. There are 2 ways of thinking it. That one is a linear progression, which thinks that the cancer cells disseminate after extensive expansion of the, so primary tumor needs to be a particular size to then the cells go out. But on the other hand, people have shown that if that theory is called parallel progression, where people have shown that actually cancer can progress, metastasis can progress in parallel to the process of tumorigenesis. So the tumor is growing and the cells in between that progression can come out and cause metastasis. So this is a very debatable issue. A lot of people generally talk about linear progression. A lot of people think, no, it's parallel progression. And we have, people have shown that the both things actually exist, but we don't know how and why and things like that. But we're still working on it. Now, let's talk about a little bit about cancer staging. So when we go to hospital for a cancer patient, they do biopsy and they give a report. And it calls that, oh, this is stage. The doctor says, this is advanced stage. This is primary stage. This is stage 4. So what are those stages? So this is a stage for the clinician to know, or the doctors to know what treatment they want to give you. So there are different kinds of staging system, but the one which is best or world over used is called TNM staging system. Now I will explain what TNM is, but remember that TNM is not used for all the cancers. The brain and spinal cord tumors or blood cancers can be defined in a different system than TNM. But what it gives, what it tells you, so it's basically, it says that where the tumor is located in the body, what cell type it is, the size of the tumor, the cancer is spread to nearby lymph node, so it's trying to move out of the tumor or not, whether the cancer has spread into different parts of the body, or tumor grade even. So let's see what TNM actually says. So TNM, the T, actually the T, so there are basically three different words. The T stands for tumor. So now, based on the tumor size and the volume, you can call it from tumor T1, T2, T3, T4, like that. So T4 is bigger tumor than the T1. Then we have N. N stands for nodes or lymph nodes. So when we do that, when the cancer moves out or the metastasis start happening, it first goes into lymph nodes, which is nearby the tumor. So if it is already in the lymph node, we call it N. So N can be 0, so there is no, it is not going out of the tumor. But then we have N, 1, 2, 3, like the tumor. So the more advanced it is, so we will see that there is a more increased number in the side of the N. Then we have the M. M means metastasize. So it's already moved out. So, and it can be different again. It can be M0, M1, M3. So like this, like T1 means tumor is very initial stage. N0, no lymph node. MX is unknown. So they don't know about if it is metastasized or not. Whereas this one is T3. So the bigger tumor, it has moved into N1. It has moved into lymph node, but we still don't have metastasized. So this way. Now, this next few slides, I will talk about the genes of cancer, what causes cancer and the different kinds of genes. So first to start with the proto-oncogenes. Now, I will explain you in a different way. So this is a car, okay? And in the car, the oncogenes or the proto-oncogenes are the genes that control the accelerator. So basically, you press it, it moves, you leave it, it stops slowly. But the proto-oncogenes are the initial form of oncogenes. That's why the proto terms is added. So proto-oncogene. So when the proto-oncogene is mutated or is gone, then it's become an oncogene. So when the basic function of proto-oncogene is disrupted and or mutated, it becomes an oncogene. Now there are more than 40 different proto-oncogenes people have identified, and people have shown they have different functions like stimulating cell divisions, inhibiting cell differentiations, or preventing apoptosis. So basically, for proto-oncogenes to become an oncogene, you only need one mutation. So one mutation, and it's good enough to accelerate the process of carcinogenesis or for the cells to become carcinogenous. Now, what are the mechanisms? So in terms of cancer biology, we call it the proto-oncogene conversion to oncogene is a gain of function. So gene has gained some function or role which is not identified earlier, or it becomes more powerful. Now, there are three different kinds of process. One is point mutation, so single mutation in that gene body or the DNA is enough to make that proto-oncogene to become an oncogene. On the other hand, the localized reduplication. So probably a particular segment of DNA has been duplicated, and hence you have now more than one copy of that gene, leading to overexpression of that encoded protein. So there is more protein in the body. Then we have chromosomal translocation. So what happens is that you have a different gene in a different chromosome, and now during translocation, that part of the chromosome is now inserted in a particular different chromosome or a different place of the same chromosome, where now it is under some different promoter, which actually is driving the gene expression. So now you have more of that gene expression going on instead of the earlier version. So now this is very famous few examples of proto-oncogenes. So SRC was the first oncogene ever discovered. It's basically Rous sarcoma virus. So again, virus. So, and SRC is basically protein kinase. Then we have HER2. HER2 name is very important for breast cancer biologists. HER2 has a large impact on breast cancer outcome and is generally amplified in more than 30% of human breast cancer. Then we have RAS. RAS is one of the famous genes in probably world because of its oncogenic property. And it's a master regulator. It can control the transcription of different kinds of genes. Then we have MYC, another big master regulator of gene expression. And then we have HTERT. I will talk about HTERT later, so I will not discuss that part. Then comes tumor suppressor genes. So tumor suppressor genes, again, is a kind of gene which basically, in a good way, suppresses tumor. So naturally, what we want that gene to be gone to form cancer. And exactly that happens. So here, I can tell you that in the same car, the genes are basically nothing but the brakes. And you know, like in a car, there are two brakes. One is hand brake, one is normal brake. So two. Similarly, for a cancer to happen, you need two mutations in the both copies of the tumor suppressor genes. If you don't have both copies mutated, probably you will not have the cancer. But sometimes it happens that someone in your family has already a one mutated gene copy. Then in your lifetime, you gain another gene mutation in the other part, which drives the cancer now. So basically, tumor suppressor genes compared to the proto-oncogenes is called loss of function, not gain. So they lose their function and hence they cause cancer. And you need both copies to be inactivated. So I already told you that a mutation in a tumor suppressor genes can also be inherited. So you can get it from your mom and dad. Now, the functions of tumor suppressor genes. People have shown that they similarly like proto-oncogenes, they have different functions like inhibition of mitogenic signaling, inhibition of cell cycle, basically in everything like growth of a cell. And then we have stabilization of genome and DNA repair factors. They can all make the tumor suppressor genes. So these are the 5 categories we have for tumor suppressor genes. Again, from the function, people try to divide them in different categories. We have cell cycle regulators, so they basically regulate cell cycle. Then we have cell proliferation inhibitors, which actually inhibit the cell proliferation. So when you mutate them, they cannot block cell proliferation, so the cells grow. Then we have proteins, so pro-apoptotic proteins, which promote apoptosis, cell death, but once mutated, they cannot do that, so the cells grows. These are basically different kinds of tumor suppressor genes. We have the famous tumor suppressor genes. P53, probably a lot of you people know. It's a very famous, world famous gene. Then we have retinoblastoma, another famous gene. We have BRCA1 and BRCA2. So BRCA1 and BRCA2 are very well known for their effect on breast cancer. So 5 to 10% people having breast cancer having mutated BRCA. And people actually target drugs to treat BRCA mutations. Now, then we have APC, which is a primary cause colon cancer. And then we have PTEN. PTEN is an interesting gene because it actually increased the risk of women having breast cancer almost 85% in their lifetime. Now, the gene basically works in cell growth or cell adhesion. So they bind cell together. Now, if you lose that protein, they start to go out and hence metastasize from the tumor. So now, so up to this part, I talked about basically basic cancer biology and how they are important and what are the basic functions and stuff like that. Now I will move into the second part of the talk, which is telomere biology and DNA structures. So during my PhD, we were in Shantanu's lab was interested in looking into G quadruplex, which I will talk later. And I was working on G quadruplex, and G quadruplex is found in telomeres. So I was interested in telomeres. So I thought it's an interesting, fascinating biology, but not a lot of people work up on it, because telomere for people is basically not interested because it doesn't directly target a drug, but it has immense importance in biology. So let's see, what is telomere? Now, telomere is basically a concept which has been conceived before the discovery of even double-stranded DNA or DNA double helix. So what happens? I don't know like how much you people know about DNA replication, so I will give you a small introduction. During DNA replication, you have the replication fork. So generally, the DNA polymerase, which synthesizes DNA, moves in this direction, and hence it creates new strand of DNA. On the other strand, which we know about Okazaki fragments, DNA polymerase, as I mentioned here, that the problem with any like DNA polymerase is that they cannot start DNA chain by themselves. So they need something to add the new bases on. So you need more bases, and you need a strand to start that. So what happens is, because biology is interesting, the cell developed a mechanism. So they actually developed another protein which we call primase. It creates RNA primer, which go and sit there, and the DNA the polymerase can now add on. The problem is, when it does that, because the replication fork is moving on this direction it is closing down. So there is not much space. So it stops after going a little bit. Then it needs to do that again. So it needs another primer to do that. But on this strand, because it's moving in this direction, they don't need another more primer because it's already opening. But here it's not opening anymore. So every time it opens, it adds a primer and it does the synthesis. So in this way, what happens? You have this discontinued DNA strands and what we generally call Okazaki fragment. Now, the problem here, I will explain you this way, that when that happens, the primase is basically synthesizing this, it's going on this way. After the synthesis is done of new DNA strands, the RNA primers are moved. So they take out the RNA primers. And then another DNA polymerase comes and synthesizes this gap, and then ligase, another enzyme, comes and join them together. So it becomes a continuous strand, now a new continuous strand. But the problem is, when it does that, the very end of DNA, you have a RNA primer here. When you remove that, there is no more DNA to fill from this end. So you leave a gap. Now, this gap cannot be there or should not be there. Or if that gaps exist, what happens is that your DNA basically, you lose genetic material, you lose genes from that part of DNA. So the nature has evolved in a way that what it did is very interesting. It put kilobases long of DNA fragments there, which are not necessary generally. So a same DNA sequence repeated over like 100,000 times to make these ends protected. So basically, if you lose them, it doesn't matter for the cell. You can lose them. They are junk in that way. Now, those ends are termed as telomeres. So both ends you have that, because when it is doing this, on the other hand, you have, at the very end, you have one primer. So the other end is having a gap that too. So both ends are having telomeres. Now, what is telomeres? So this is a metaphase spread. Probably all of you know about metaphase spread. Now, if you do a metaphase spread and you try to use DNA sequence, DNA probes of that same sequence of a telomere to know, you will see that all of these ends are lighted up. So basically, every chromosome has 4 telomeric ends, like 1, 2, 3, 4. So in total, we have 96 of them. So basically, they are repetitive DNA sequence. The word telomere means telos. It's a Greek word. It's called telos, the end, and meros, the part. So Together, they stand for telomere, or end part of your DNA. Now, telomeres was discovered 1930 by Barbara McClintock, again, plant biologist, corn. So she was working on corn, and she figured out that corn chromosomes sometimes stick together. So she was figuring out why. And then they discovered telomere. So I will talk about why they stick. So the basic human and all vertebrate sequence for telomere is TTAGGG. These 6 bases are repeated 1,000 times at the end of the DNA, and hence they make a repetitive sequence, what we called. Then this repetitive telomeric sequence, due to this sequence, that DNA repair system, I will show you a few slides that how DNA repair sequence don't repair them. And then people also believe that these telomeres, every time the cells divide, as I showed you from the end, you lose some DNA from here, which is still you are losing, but you are losing some non-essential DNA, which you don't need, your cell doesn't need. But every time you divide, you count, the cell measure how much DNA you are losing. So if you are losing like, suppose it will generally lose almost like 100 bases approximately, depending upon the organism. Every time you lose that, the cell counts it. And once that counting reach a critical counting point, the cells stop divide. It just don't divide anymore. So that's we call it a mitotic clock. You count every time you divide. Now, telomere structures. So telomere is a fascinating DNA structure in terms of its structure, because it has a lot of different kinds of DNA structure. So first, let's see. So it has a 3' overhang. That means if you look at this picture here, you see that the 3' is longer than the 5' because you don't have the RNA primer here anymore. So it's always smaller. So it basically, you have a 3' overhang of DNA, single-stranded DNA, just like this. And then generally telomeres are replicated by the normal replication machinery. Nothing new there. But there is a protein called telomerase, or there is a method of homologous recombination-based pathways which elongates the DNA. I will talk about it in a couple of slides later. And hence, every cycle, it increases the 3' end, and the replication machinery fills up the lower strand. And it's generally, telomere is bound by different proteins like histones and normal other proteins, but it has a core protein structure. So there are 6 proteins discovered in different time points between 1998 to like till people are still discovering telomeric proteins. There are 6 proteins. One is TRF1, which is telomeric repeat binding factor 1, because it's been discovered first, so 1. Then discovery comes for telomeric repeat binding factor 2, so the second one. Then we will have TPP1, RAP1, TIN2, and POT1. So this 6 proteins makes a complex and go and bind to the telomeric DNA only. Although people are right now figuring out that there are more, they can bind to other places of DNA too, but still, primarily they bind to the telomeric DNA. And they binds like this, just like histones, equal distance. And then we have the telomerase, which works on the single strand overhang. Now, let's talk about telomerase. Telomerase is a fascinating enzyme. It's fascinating because it's the only reverse transcriptase we have in our body cells. So basically, it's a hollow enzyme. Means there are 2 components of this enzyme. One is the RNA component. So it's an RNA-based reverse transcriptase. That's the interesting part. It's an RNA-based reverse transcriptase, but it synthesizes DNA. The other part of the protein, the other part of the complex is the protein itself, which we know as HTERT. And so the RNA part is HTR, human telomeric RNA, and the HTERT, which is the enzyme. Together, when they come together, they make the hollow enzyme. Now, the hollow enzyme, basically this enzyme was discovered back in 1985. Now, interesting part is the whole telomere biology is ruled by women. So they are like all the big scientists in this field are basically women. So it's a woman-dominated field, which is interesting and which is very nice, good to. And I interacted quite a few of them, and they are all nice ladies. So what they discovered back in 1985 by these two ladies, Carol Grader and Elizabeth Blackburn, they actually won Nobel Prize for this back in 2007 or 8 for discovering this enzyme. So it's a ribonucleoprotein complex. Now, how it does that? So telomerase is basically active in all germ cells, in vitro immortalized cells, cancer cells, stem cells, and white blood cells, but it is not activated in your normal body cells. So when you are born, there was telomerase. Over your age, you lose telomerase activity. So your cells, that's one of the reasons you age, your cells age. So your mature body cells doesn't have telomerase at all. Now, the interesting part about telomerase. So again, I would say the beauty of biology or beauty of evolution. So telomerase, the TERT protein itself, actually is just like any other viral polymerase. Viral. That is the interesting part. It's not human by origin. So it's basically a viral. It's basically a viral origin. So if you see a retrotransposon or like a HIV virus reverse transcriptase, the telomerase gene here has exactly same polymerase structures, protein structure, like the HIV virus here. So by core, it's a viral polymerase. So how it came into human body? There are different theories how it came. But one of the theories is somebody in the very earlier system got infected by a virus which has this protein, and the cells figure out that this is very beneficial for me. So they kept it inside the genome. So it's integrated. And because it's so important, cell never rejected. It made it its own. So they have it for. So that's how. So that's another point that why telomerase is so interesting protein. It's a viral protein. Now, how telomerase actually elongate telomeric DNA. So what happens is that this is the RNA sequence, which is the HTR in telomerase. And this is the protein, the big round thing. So it goes and it binds to the 3' overhang of the telomeric DNA, the TTAGGG. Now, it goes and it's bind, and then it starts amplifying that DNA, the single-stranded DNA. So it becomes longer. Now, during the next replication cycle, the RNA primer can go and bind to this end, and hence the DNA can be elongated. So you have longer DNA now, which from the initial stage, which is shorter. So this red part is newly synthesized DNA. So generally, that's how it elongates DNA. But once you are adult, the telomerase stop working. So you'll start losing this, and the cells, once it reached a critical mass, it loses. The cell loses the potential to grow again. Now, the telomere theory of aging. Telomere is fascinating because a lot of companies are putting a lot of money in telomere biology because of the aging concept. So what is aging concept? So basically, potentially, immortal cells maintain telomerase activity and can divide indefinitely. Simple, right? You will elongate the DNA, so the cells will never reach the phase where it stops dividing, so it will go on. Now, but generally our body cells have a limited replicative lifespan because we don't have active telomerase in the normal body cells. So it shortens the telomere. Now, this actually leads to the replicative senescence. When you reach a critical length of telomere, the cell doesn't want to grow anymore. Now, this concept is called Hayflick limit. It's been discovered by a very famous biologist. He was a doctor, and he discovered this. So Hayflick limit is, in a simple term, is that the number of times a normal human cell population will divide before cell division stops. So once you reach that critical number, the cell will not divide anymore. That's Hayflick limit. And if you take your body cells out from your body, put it in a culture plate, like you grow cells, you will see after 10 or 15 divisions, those cells will not grow anymore. They will stop growing. That's half-life limit. But a cancer cell doesn't have a half-life limit. It can grow beyond that 10, 15 divisions. So it can, like, one of the cancer cells which we know probably is the HeLa cells. We use a lot of HeLa cells in our cancer biology work. HeLa cells grow indefinitely. So it's been isolated from a tumor and it's still growing. Interesting. So telomere, now let's talk about telomere shortening, I would say the interesting part of telomere biology. It's called telomere shortening, cellular immortalization in cancer. So how telomere is related with cancer? Now the normal cells here, you can see that it has, when you are born, it has 10 to 14 kb of telomeres. Simple. Every division you are losing some telomeres. So now after 50 to 80 cell division, the telomeres reach a critical point. And so the length at that time is 4 to 7 kilobases of telomeres. And it leads towards senescence. So now the cells is thinking that I should die. And it goes 30 more cell division approximately after reaching that phase. Then the cell feels, oh, I'm at a critical telomere length. I should not divide anymore. I should stop right now. So that critical length is 2 to 4 kilobases of telomeres. Now the interesting part. So in cases of normal cells, they will choose this pathway of cell death and they will die. But in a cancerous cell, these cells will choose that, okay, I don't want to die. So they will reactivate telomerase or an alternative lengthening of telomere mechanism and start elongating their telomere up to that critical stage, which is 4 to 10 kilobases. So they are back to here again. So they maintain that length for the rest of their life. They continuously do that with the help of telomerase. Hence, so you can see here that normal cells, you have the yellow telomeres here. It's reached a critical mass, so the cell stops growing and leads to cell death. But in a cancer cell, it keeps maintaining that telomere length. Hence, the cell genetic makeup changes and it becomes a malignant tumor or a tumor. So this is the part of telomere biology which a lot of biologists are actually interested in to understand this. Now, telomerase and cancer. So more than 90% of human cancer types are telomerase positive. More than 90%. Actually, it's close to 95%. Now, somatic mutations in the proximal promoter. So every gene has a promoter region, right? So the promoter region generally take a lot of mutations. So the promoter region of telomerase genes is the highest mutated part of any non-coding mutation in cancer. So it doesn't, so they have the maximum amount of mutation in the telomerase promoter. People still are looking for why it is so, but nobody knows. So, however, there are another mechanism, which is homologous recombination mechanism, where the cell don't use the telomerase-based pathway. It uses homologous recombination-based pathways to elongate the telomere. Now, there are different kinds of like mutations or engagement of telomere, alternative splice. So there are different forms of telomerase which can form, but the basic function remains same. The cancer cells want the telomerase to be active to live forever. Now, as I said earlier, why telomere was important in terms of telomere biology, why it's important in terms of chromosomal biology. So generally, when we have a double-stranded DNA like this, there is a double-strand DNA break, which I talked about earlier, and you have now a breakage in the two strands of DNA. So generally, DNA repair activity happens, it repairs the DNA, and now you have a finely repaired DNA, which is green. So good for the cell. But the interesting question is that's why we need the research. I would say that research is interesting because we ask questions which are already there, but nobody sees it or nobody understands why it is there. So that's research. So people thought, okay, fine. What happens to the end then? That was Barbara McClintock. She thought, okay, this was the end. So what happens? So there in cell, you have multiple telomeric ends, not one. You have like 96 of them. So if all of them are like this, so if a DNA repair activity happens, recognizing this as double-stranded break, which is natural for them because they don't understand it's a telomeric end or it's a normal DNA end. It's just DNA end for them. What will happen? They will come, they will repair it. Now, what will result is that fused telomeric ends or fused chromosome. Fused chromosome means now you have genomic abnormality or genomic instability, which will lead to actually cancer. So that's why we call it dilemma of repairing the end. So how you repair the end? Now, this is one of my favorite slides. We call it all's well that ends well as a loop. So if you hide that particular this end in a loop inside you. So now the DNA damage repair proteins don't identify that there is a break because the ends are hidden inside a thing, just like a rope. So there is no end. So the DNA repair proteins don't recognize it as a damage. But the problem was when this hypothesis was discovered, people thought, but how can this be true? How this is actually can happen? Because that is so complicated. So here comes my first postdoctoral guide or boss. His name is Jack D. Griffith. You people probably have seen a lot of his images if you ever read Cell by Cooper. In that book, all the electron microscopy images are actually from his lab. He used to be Arthur Kornberg's student, and he has excellent, interesting way of thinking biology because by degree, he is a MIT physics graduate or physics PhD. So before coming into Arthur Kornberg's lab, he never did biology. So what this gentleman actually thought was very interesting. He said, okay, let's try to see it. So at that time, 1999, electron microscopy was advanced, but not today's advancement. And he developed a way to make DNA out from the cell and look under the electron microscope. And amazingly, he saw that this structure, he saw that the DNA ends are folded into a loop like this. This is from the pea plant. So it has much longer DNA. It's basically, and if you look at this image, you will figure out that this small round circle here, it's 3 kb of DNA, 3 kilobases of DNA. This thing is 22 kilobases. Like this thing is 18 kilobases. They are huge structures and they actually form inside the cell. And this particular paper is a landmark paper in biology. It's a super landmark paper actually, because this has been cited more than 3,000 times. So when I joined his lab, okay, one picture is not working. So when I joined his lab, he was trying to figure out a way, the whole field was actually trying to figure out a way to mimic telomere in vitro. Like can you synthesize long telomeres just like we have in our human cells, but in a test tube? So I used biochemistry to actually synthesize first the longest double-stranded telomeric DNA in vitro in a tube. And when we use the same mechanism they use inside the cell, we actually generated loops. So this is basically in test tube generated telomeric DNA, and these are not small, these are like 40 kbs of DNA, and they actually can form the loops. Exactly. Now, let's see. Now, I think I like to be interactive, but still I will question. Question is, up to this point, did anyone think why the nature has evolved into a sequence, not random, like why it is for every vertebrate or every human, it is TTAGGG always. Not like TTCCCCT, nothing. It's always TTAGGG. So why that sequence is so important for the cell? And why cell wants to maintain that sequence? So, as I say, it's elementary. Biology is always elementary. That's what fascinating about biology. It's always elementary. So the elementary part is, you can see that all this sequence from different things like humans, mice, Xenopus, even plants, all they have common is this G. They have at least 3 Gs like this, or even 4 Gs. Sometimes they have more like T, GG or variable, but there is always G. Why it is so G-rich? Now, here comes G quadruplex. So this final part of my presentation, I will talk about a DNA structure which now people know is all over the genome. There are lakhs of G quadruplex inside your genome, and they've actually formed inside your genome. You can see them. What happened is, like, a lot of biology happens by accident. A lot of discovery actually happens by accident. So Everbang, this Scandinavian fellow, he was a biochemist. At that time, they used to call them chemist, not biochemist. So this chemist, he was working on guanylate acid, and he had a beaker full of guanylate acid solution. He kept that overnight because he forgot. Naturally, as a scientist or as a researcher, we forget things because it's only a single brain. He forgot that solution overnight on his desk. The next morning when he came back, he saw that whole solution is not solution anymore. It became a gel. It was gel. So he thought, why it became a gel? But he never couldn't explain why it was gel. But then, after double helix discovery, Gellert in 1960, now having crystallographic method, tried to analyze that gel, and he saw something amazing. He saw that the guanylate acid can form a secondary structure, and that structure he called G quadruplex. So basically, what is quadruplex? So if you look at here, they are like, suppose this is the telomeric sequence, TTA, GGG. So you need 4 repeats of this sequence, 1, 2, 3, 4. Now this 4th sequence then fold. They fold like this. They can fold like different way, but one of them way is this. Now during this folding, one of each of these G's, 1, 2, 3, 4, they organize themselves in a planner, like a simple, just like a, suppose like this, like a page. Then you have another page of G. Then you have another page of G. Now, when each of this plate of G is called guanine tetrad, because 4 guanines are there, so tetrad. When multiple of these tetrads stack themselves, so they organize themselves one upon another like this, you get G quadruplex. Now the most interesting part, quadruplex, when they form, they are more stable than your DNA double helix. So if they have formed, you cannot open them up easily. It's very hard. If you take this sequence and it forms a double helix, now if you open it up, it forms a quadruplex. It will never go back into double helix by itself. You need other way. And what happens is that in our body, we have potassium ion. Potassium ion generally goes there, other than there, like potassium, sodium, lithium. They go and they stabilize this G tetrad. So they are so stable, they don't open up easily. Why it is important? So as I said, there are 376,000 potential quadruplex sequence in your genome, which people have shown they are there. And they're important because they control biology like transcription, replication, even at telomeres, or even during translation. And not only that, that it can be DNA-based structure, it can be RNA-based also, because RNA also have G. Wherever there is a multiple repeat sequence of G, there can be a possibility that quadruplex can form. Now, so I will not go into details of quadruplex in whole biology. I will just focus on telomere biology. So when I was in Jack's lab, there is a long-standing question that, okay, this definitely is entered in a loop and can be protected, but once it opened up, how it is protected? Why the cells is not detecting it as a single strand? So people thought that probably because this is TTA-GGG repeats, it can form quadruplex, and they showed it. But the problem is this is 150 to 300 nucleotide long. And for a quadruplex to form, you need only 24. So there is a possibility that there is multiple quadruplex like this. But the problem is nobody knows how they are organized, if there is actually multiple quadruplex. So the question was, the argument is probably they are organized in a bead on a string. So like mala, it's like a putiru mala, basically. Putiru mala to যেমন পুঁতিগুলো সাজানো থাকে, it's just like that. So what I thought will be an interesting way to see quadruplex is electron microscopy. So in my paper back in 2018, I actually showed that, okay, this is the series strand, which is the complementary strand. Or this is a mutant sequence where you don't have continuous G, you have a T in between TTA, GGG. But when you take the telomeric TTA, GGG repeat, it organizes itself in exactly bead-like structure on a long DNA. And we also showed that these bead-like structures are actually 4 quadruplex coming together and forming one of those beads. So each of those beads has 4 of those quadruplexes. So they are basically 96 nucleotide long. And that's why the end is more than 96 always, generally. So what is the biology or importance of quadruplex in terms of drug targeting in cancer? So this is what is happening generally. You have the normal TTA-GGG overhang. It shortens, the cell division stops. But in a telomerase active condition, this overhang is particularly elongated and your cell gets unlimited proliferation. So what happens is that if we have this overhang, which is forming G quadruplex, now we use a ligand. So basically it's a chemical stuff which can go and never let this quadruplex to open up. So it will block, it's in a quadruplex condition. So what happened, telomere cannot access this part anymore. And hence telomerase cannot extend this part. That becomes, so that we call them G quadruplex ligand. So hence leading to a telomerase inhibition, and that will lead to cell division. And the cancer cell will have a negative effect. Okay, so these are all the telomerase inhibitor based on anti-cancer drug strategies. But you see that there is a G4 DNA stabilizers, which is G quadruplex, we call them G4. And they are like BRCA19, and there are Piper, and there are other where actually people use them to show that they have their restrictions, and people are still trying to come over those restrictions because everything, the inside test tube doesn't work exactly like outside test tube or inside our body. So we have issues with those. Now, I want to finish this off by thanking all 3 of my mentors. Shantanu Chaudhary. Shantanu basically helped me starting my career, and he's still immense support, moral support and scientific support for me. And I worked in IGIB, it's in Delhi. And then I moved from IGIB to Professor Jack Griffith. He's an excellent mentor and excellent guide and excellent human being. And I worked with him in University of Chapel Hill in North Carolina. And then my current boss, Professor Paul Doetsch, and I work in NIH-NIEHS. It's in the same place in Durham, North Carolina. And here I am currently doing my DNA damage repair biology-based research. Thank you. I can now have the questions.
Thank you, Dr. Paul. It was an excellent presentation. We are mesmerized by the way you presented this topic. And lastly, when you showed your slides, the work that you really did, it was fascinating. And I must say that it was a very a webinar which had a lot of facts, but you had mixed a lot of wit and humor also to it. So that made it even more interesting. Like I really liked your quote, "All's well that ends well." As it, I mean, it was so interesting. And this journey, we have a lot of questions. And Dr. Moushumi Dash and myself, we have selected some questions. But you know, maybe we don't have the time for all. So it's coming on a screen. Can you see it, or did one? The question on your screen. Yeah. So if you can read the question and give the answers.
Okay, so one of the question, I think Revonto. So the question is cancer a constraint in case of immortality or anti-aging research? Because these two are quite antagonistic to each other. Exactly. You are perfectly right. So basically, cancer যখন হয় আমরা basically cancer biology একটা important part হচ্ছে immortality, cellular immortalization. And the problem actually is that a cancerous cell generally is always immortal. But an immortal cell should not be always cancerous. So there are people যারা এই নিয়ে কাজ করে, তবে one point probably a lot of people generally ask me that আমি কিছুটা example দিয়ে বলছি, যেমন one of the cancer theory is that আজকের ডেটে ক্যান্সার occurrence বেড়ে যাচ্ছে, তার পিছনে কারণ হচ্ছে আমরা বেশি দিন বাঁচি। Your age, because of modern medicine, we now survive like 80 years, 85 years. At 85 years of your life, you are continuously getting mutations. During mutation, so more you live, more mutations is in your body. More mutations you have, you are increasing the cancer risk. আগেকার দিনে ক্যান্সার occurrence probability অনেক low ছিল আজকের তুলনায়। As for like, so it's very hard to isolate them from each other. It's very hard. You cannot, almost. But maybe in future we can discover a way যখন আমরা ক্যান্সার সেল ভার্সেস immortalized সেলকে আলাদা করতে পারবো। But definitely it will be a tough job, but yeah, it's not impossible, at least. So they are basically the question is, what is the difference between apoptosis and programmed cell death? There are so it's basically a similar kind of term, but in terms of deep biology they are different. It's basically programmed cell death is apoptosis, but it has another part of than apoptosis too. So yeah, it's a more of a subclass of programmed cell death. Yes. So the question is, as per the sequence analysis, how can it be directly concluded that the telomerase activity evolved from the retroviruses in the co-evolution of humans? Viruses, in the co-evolution of human viruses, what is your opinion on reverse case? I think the question is, like, as per the sequence analysis, how can it be directly concluded that telomerase activity evolved from the retroviruses in the co-evolution of human viruses? Okay, so okay, I think the question, one of the answer will be, when we have telomerase is right now incorporated in human genome. Okay, but the cell needs to understand that it's important for them to have it inside the cell or keep it inside the cell, so that they can use it. Now the problem with other virus particles, like, so HIV, suppose HIV has the same thing. When it infects the cell, it uses the RNA template and it modifies it and synthesizes its own system. But that is also reverse transcriptase. So it's exactly working similar way than with like telomerase. But the cell, human cell or the eukaryotic cell, they recognize that this thing we need, and this thing we need because this thing is helping us to survive. Actually, if you do sequence analysis, like whole genome sequencing, which I did actually, you will see a lot of human genome has sequences which resembles virus sequence, which is interesting. So people think that a lot of time, though, when those virus infect you, they leave traces of DNA or of their original DNA inside your human genome. It generally stays as junk, but if your body needs it, they can use it probably. But yes, it can evolve with your DNA over the time, and probably in over like next 100,000 of years, we'll have some different viral protein which is helping us to survive. Yeah, that's biology. Yeah. So the question is, you gave a great insight about stages of cancer. Can you give a explicit knowledge on what are the factors which can accelerate each stages with regards to its biochemistry. Okay, it's going to be tough. The problem, it is tough because every stage of cancer. So let me explain a single example. MYC. MYC is a well-recognized cancer protein. Everybody knows about MYC. Okay. The problem is MYC has two different functions. People have shown that initial stages of cancer biology, it was a tumor suppressor, which I showed you. It then, once mutated, it can promote growth, but MYC can also drive metastasis. So MYC, being a tumor suppressor, it can also promote metastasis. So every stages of cancer, a same protein can behave differently. Same happens to TGF-beta. It's supposed to stop the tumor, but in advanced stage, it promotes basically metastasis. So it's very hard to identify each of them in a single, or say that, okay, this protein will only work in this stage. No. They network, they are much well connected than today's human beings. They work in conjunction with each other, they talk to each other, and if needed, each of them can have multiple roles in multiple stages of cancer progression. You cannot define them based on a single role. Yeah. So the next question is, to what extent has the mechanism of carcinogenesis been conserved throughout evolution? So I can talk about animals. So like dogs or other animals, they have osteosarcoma. It's the same way the tumor forms. On the other hand, osteosarcomas have been reported in humans with younger human beings, where they are more active. So it's been conserved in basic tumor, when we talk about basic process steps of tumorigenesis, but with evolution of more complicated systems, it always evolved with that. So you have more factors contributing to it. You have more mechanism to stop it in higher organisms. But so the problem is, the higher you get, the complicated it gets. The lower ones have easier biology to understand, but the higher ones are much more complicated. A human cell is a network. That's why we call it network biology, where you use genetic idea of genomics, proteomics, transcriptomics, all together comes, and you try to understand how all of them are actually working together. That's where it's become more complicated for human biology. So PTEN gene is also present in Arabidopsis, zebrafish, human, and other animals. What is the functional difference of PTEN genes? I cannot talk about plant because I left plant biology quite long back. But in animals, I can talk. In animals, PTEN is basically a cell growth promoting gene, and also it's an addition molecule. So it connects two cells. Once they get mutated, the connection is lost. So now those cells can move out from their way. So basically, it's bind you to the other cell. Once you lose the binding, you can move and go to anywhere. So that's how basically PTEN works in animals, at least. If a cancer cell is in dormant stage, how can we tell it is cancerous? It's very hard to detect a dormant cancer cells. People are doing whole body scans to identify, or they are using different kinds of probes which can go and probably find those kind of disease cells inside your body. The problem is with that kind of approach is that you cannot do that unless you know exactly what to target, because probably the same protein is present in your normal cells and in your cancer cells. So the probe should be targeted towards that particular cancer cell or that particular protein which is altered in cancerous condition. So, but people are, that's one of the diagnostic part of cancer biology people are working. I personally know people who actually does here in UNC, and they actually tries to figure out what is the best way to identify dormant cancer cells. But yeah, probably in next 10, 15 years, people will confirm that that's the way you work. But now there are ways to detect them, but they are not very conclusive. If we cut down the source of food, does still the cancer cells will show immortality? Oh yes, they will, definitely. Because the way cancer cells survives, they can survive on very low energy. Their whole metabolism changes. Because just for example, in a tumor, so tumor is a mass, right? So the cells on the surface of the tumor doesn't have, so they are exposed to nutrients, air, and stuff like that. But the cells which is in the core of that mass, they don't get those things very easily. So they are not exposed to that much of oxygen or food, but they still survive. And they are more dangerous than the superficial cells. Because they survive in that condition, in so low metabolic state, they adapt to an alternate metabolism state. And that alternate metabolism state helps them to be alive. So yeah, they can serve the alternate metabolism state they can survive that. So there is actually an option for them. Which chemical property of guanine calls the quadruplex? Okay, so why not adenine chosen? Okay, so the property is probably you already know, it's called hydrogen bonding. So the hydrogen bonding in case of guanine, if I can show it probably. So I will share the slide so you will see that guanine, all those 4 guanines are actually connected with hydrogen bonds, and that hydrogen bonding is discovered. It's not normal hydrogen bonding. The person who discovered it is known as Hookstein. So we call it Hookstein hydrogen bonding, and it's a special way. If you look at the organization of adenine, if you try to fit that organization in adenine, you cannot fit it structurally. That's why guanine is chosen to do that hydrogen bonding. A lot of people tried that, but it just don't fit. That's how the whole system has evolved. You cannot fit adenine in that hydrogen bonding. It will not fit and stabilize enough. But the interesting part is there is a structure which forms on the other strand, so the guanines, when they form, it's called quadruplex. On the other reverse strand, you have C richness. Naturally, G, you have C on the other strand. Those C-rich strand can form a structure similar to quadruplex. We call it I motif. I motif, the problem with I motif is that it needs a different pH than the physiological pH. So they're very hard to form. So they generally don't form, but they can form, chemically speaking. So yeah, adenine just don't fit like the guanine in the chemical structure dimension. What are the parameters that determine a competent immune system that gives a complete response to cancer after immune induction? I am not an immunologist, so I will clear that first. But when it comes to cancer biology, immune systems or immune parameters really doesn't hold that strong because cancer cells evolve. So every time they evolve, they have escape mechanism to escape immune system. So simple, like just for example, when those cancer cells travel through blood vessels, a lot of those cells get killed, or even they walk through immune systems like lymph nodes. They get detected, they get killed, but very few of them develop mechanism through which basically they escape that. They automatically within very short term they can evolve. So just for example, if you give a person a drug to treat a cancer, definitely you will kill most of them, but then the cells start to evolve. So they evolve in terms of drug treatment, they evolve when it comes about immune suppression. You cannot just define cancer and you just cannot treat cancer and say, oh, we treat cancer once for all. No, it will come back, it will evolve again. So the idea is to understand the mechanism, how it evolves, so evolution is a very integral part of cancer biology. It will evolve. So the parameters of immune systems are not exactly fitted when we talk about immunosuppression or in terms of cancer biology. It will hold true up to a limit, but it will again come back and it will evolve. Is T1N2M2 both more difficult stage than T3N2M0? Is the size of tumor metaphor? Okay, I already told you that. So T1, let me explain it this way. T1 is tumor 1, N2 is nodal 2, and M2 is metastasis 2. In terms of a doctor's perspective, T1 N2 M2 is more deadlier compared to T3 N2 M0, because M0 means there is no metastasis, so it's not spreaded out yet. But when you have M2, that means it has already spread out. So now you cannot target that particular tumor with your drugs. You need more efficient way to treat all those metastasis conditions too. And once it is M2, it's like quite a high condition. It's not anymore very easy to treat. So yeah, the first one is more complicated to treat than the second one. What is the basic difference between apoptosis and necrosis? Yeah, so I told you, like, they are two different terms. So if I remember correct, apoptosis is a cell death, and necrosis is basically decaying, how you decay the cell out. You clear that system out. But they are different. I cannot go in detail of that because I don't have a very clear idea, but I know that one is disposal of the cells, one is the cell death. Due to presence of phytoestrogen in soya, it helps to cure breast cancer and ovarian cancer. How it actually cures this cancer and what is the impact of it? Okay. So, okay. Being a cancer biologist for this long time, let me talk about something. Okay. I am not a very good, person when it comes to treating cancer in terms of this kind of therapy, especially plant-based. Like a lot of people showed that curcumin, suppose, has anti-cancerous property. But the question is, how much of that curcumin treatment actually holds true in an FDA standard long-term cancer treatment? It's very debatable. Exactly like the phytoestrogens and all other plant product-based systems. The point is, you are still having a chemical. It's from soya, but it's a chemical. The point is, are those rigorously enough tested? Rigorously, the word is rigorous. Are they rigorously enough tested to be advised by FDA to be real cancer treating them? There are papers. So when we talk about curcumin, you will find thousands of papers in the PubMed, or like phytoestrogens or even different kinds of other plant-based things. Problem is, once you move into the clinical trial part and rigorously done clinical trial part, it's different. It's a completely different scenario. It's not easy that, again, even for a lot of drugs in terms of quadruplex biology and different things, there are thousands of papers in PubMed, you will find. But the problem again comes is how much they are clinically trial tested and FDA approved. That is the click part, FDA approved. Because yeah, you can get a drug and it will probably diminish in a mouse model. You will see there is a diminish of tumor. And you say, okay, it's working. But does that actually hold true for a long-term cancer therapy in human? That is the problem. There are a lot of cancer drugs which actually claims to do that, but there are very few which actually does that. So there's a difference between can do that and actually does that. Is there any role of cancer in the evolution of complexity of DNA structure? That's a good question. That's a very good question, actually. The role of cancer in the evolution of complexity of DNA structure. I don't know, actually. It's a good question because I don't know really that it can be other way around, that the complexity of DNA structure can actually impact cancer evolution. But if the cancer is actually affecting the complexity of DNA structure, I haven't really read about it anywhere till now. But yeah, that's a good question because I know the other way around can be possible. Like if you have a DNA structure which actually, like quadruplexes, they evolved. Like a plant has a different quadruplex sequence than a human. So they evolved, and they also, like human quadruplexes, they try to block protein from working on them, and they block protein from transcribing them. Hence, they actually block the expression of carcinogenic genes. But on the other way, they can also promote depending upon the context. So yeah, the DNA structure can actually affect the biology of cancer, but I don't know really that the other way around is actually can happen. But biology, it can happen.
Okay. Dr. Anirban Kar, we have a lot of questions. I mean, there are so many questions which are still unanswered, but you know that there's a time constraint. So we
Sure.
request you that since you'll be sharing your presentation, so you just
Yeah,
insert your email ID there also. So whoever is interested to get in touch with you can email you, because
sure.
and then you can take up their questions on a one-on-one basis. So I think we should draw an end to this session, the question and answer session. And I think the only thing left is the formal vote of thanks. So I think I'll go straight to that because I'll be giving the vote of thanks. So here we go. Okay, so the vote of thanks. So good evening, everyone. On behalf of the Botany Department of Gurudas College and the IQAC, I take this opportunity to propose the vote of thanks for all those who have helped in organizing this webinar jointly with the IQAC of this college. Now, first of all, I must thank the speaker, Dr. Anirban Kaur, for his wonderful talk on the understanding the complexity of cancer progression and biology of DNA structures. It was indeed a rare opportunity to hear a talk on the biology of DNA structure and the complex cancer progression process from a research fellow working in the Genome Integrity and Structural Biology Lab in the prestigious National Institutes of Health, National Institute for Environmental Health Sciences, USA. Now, cancer biology is a topic in the syllabus for nearly all the life science subjects, and this wonderful presentation today have made this topic even more fascinating. Now, in the times of COVID-19, we are all hoping that a vaccine will be available soon. But we have to remember that any vaccine or medicine is produced by the cumulative hard work of scientists and the research teams. So today, after this webinar, even if a single student thinks of pursuing research as his or her career option, I think we would have fulfilled our objective of the webinar. I must thank our principal, Dr. Moshmi Chatterjee, for all the support for organizing this webinar. I must also thank the IQAC convener, Professor Tripona Mojumdar, for her encouragement and help. All my departmental colleagues have supported this initiative and I thank them all. Now, I have to thank all the participants, both faculty members and students of our college, as well as other educational institutes, for attending and participating in this webinar. The question and answer session really became lively with all your interactions. Now I have to thank someone without whose support this webinar could not have happened so smoothly. It started almost, I think, a month ago with the finalizing the dates, making the flyers and the video, meeting in the digital studio for a mock demonstration with the speaker, and also, of course, the technical support today throughout the entire webinar. Thank you so much for your constant support, Dr. Joydeep Chandra, the librarian of Gurudas College. Now, there may be some people whom I have failed to mention. Plus, please remember that the Department of Botany gives thanks to all those who helped in the organizing this webinar. Now, Dr. Anirban Kaur, you have mentioned that you'll be sharing your presentation. It will be available in the Gurudas College Courseware from where anybody can download it. Thank you so much, Anirban, for this. Now, before I end, I must say that the fact the speaker is an alumnus of our Gurudas College makes this webinar even more memorable. Anirban, during your days, you have told us, your teachers, that you are very interested in research. You have worked hard despite several odds in chasing your individual passion and dreams. You have shown that belief in one's own passion and having the self confidence and also the hard work pays in the end. You have been and will always continue to be an inspiration for our students. Thank you so much, Dr. Anirban Kar. So with that, I think we come to the end of this webinar. It was great having you here, Anirban, and we hope we'll meet in person someday. And
Sure.
till now, in the virtual field. So would you like to add something about your college days? Because there are some questions regarding like, why did you switch over to cancer biology, your journey? A little bit, just 1 or 2 minutes.
Okay, I will take maybe 2-3 minutes. So যখন আমি কলেজ ছাড়ি বেসিক্যালি প্রথম কথা আমি কিন্তু কলেজে ফার্স্ট ক্লাস পাওয়া স্টুডেন্ট নই ফার্স্ট সুতরাং বাট আমি কলেজে যখন পাস করে বেরোই বটানি অনার্স নিয়ে আমার কেমিস্ট্রি এবং জুলজি দুটোতে নম্বর ভালো ছিল দ্যাট হেল্পস কারণ অল ওভার ইন্ডিয়াতে কম্পিট করতে গেলে ওদের দরকার পড়ে ইটস আ কিউমুলেটিভ নাম্বার সিস্টেম অল ওভার ইন্ডিয়া সুতরাং আমি যখন ওখান থেকে ছাড়ি আমি ভোপালে যাই আমি তখনও পর্যন্ত ডিসাইড করিনি তখনও পর্যন্ত যে আমি রিসার্চ করব আমার রিসার্চ করার বাট হ্যাঁ আমার এক্সপেরিমেন্ট করতে প্রথম থেকে ভালো লাগতো ফলে আমি এক্সপেরিমেন্টাল বায়োলজিতে ইচ্ছুক ছিলাম তা যখন আমি ছেড়ে বেরোই তারপরে আমি যখন আমার প্রথম রিয়েল ওয়ার্ল্ড এক্সপেরিমেন্টাল বায়োলজির সঙ্গে ইন্ট্রোডাকশন হয় শান্তনুদার ল্যাবে অ্যান্ড শান্তনুদার ল্যাবে আমি যখন ঢুকি আমি স্টার্ট করি বা পড়া আমার কাজ করা 6 মাসের জন্য ট্রেনিং ছিল অ্যান্ড আই ওয়াজ ভেরি ইন্টারেস্টেড অ্যান্ড বিকজ অফ দ্য অল ডিসকাশন কীভাবে ইন্টারন্যাশনাল সায়েন্স হয় সেটা জানা এবং শান্তনুদা ওয়াজ ভেরি পেশেন্ট উইথ মি লাইক হি ওয়াজ outstanding. He still is outstanding. So, and so after doing that, আমি 6 মাস যখন কাজ করি তারপর শান্তনু আমাকে অফার করেন যে if you want you can join my lab. And already তখন আমি ইন্টারেস্টেড ছিলাম বায়োলজিক্যাল সায়েন্স করার জন্য and শান্তনুদার ল্যাবে ঢুকে আমি এটাই জিনিস বুঝতে পারি যে biology is not defined by botany honors or zoology honors or physiology honors. Biology is everything. সো বায়োলজিতে তুমি যেমন একদিক থেকে প্ল্যান্ট নিয়ে পড়তে পারো কিন্তু প্ল্যান্টের বেসিক বায়োলজি অলদো হিউম্যানের থেকে ডিফারেন্ট বাট দে আর কানেক্টেড যেমন একজন একটু আগে জিজ্ঞেস করলেন যে P10 অ্যাজ আ প্রোটিন প্ল্যান্টেও কাজ করে হিউম্যানেও কাজ করে ডিফারেন্ট ফাংশনও থাকে দিস অ্যাকচুয়ালি মেক মি টু আন্ডারস্ট্যান্ড দ্যাট ইটস মাচ মোর বিগার পিকচার দ্য পিকচার ইজ বিগ সো অ্যাজ আ রিসার্চার সিন্স শান্তনুদার ল্যাবে আমি জয়েন করার পরে টিল টুডে আমি বিগার পিকচারটা দেখতে পছন্দ করি আই ডোন্ট ওয়ান্ট লাইক আমার কাজের কিন্তু একটা পার্ট যেমন পিওর বায়োলজি একটা পার্ট কেমিস্ট্রি ডিএনএ ড্যামেজ বায়োলজি ইজ পিওর কেমিস্ট্রি নাও অন দ্য আদার হ্যান্ড যখন আমি ইলেকট্রন মাইক্রোস্কোপে বসি ইটস বায়োফিজিক্স সো ডোন্ট দ্য অনলি থিং উইচ আই ক্যান সে ইজ দ্যাট ডোন্ট বাউন্ড ইয়োরসেলফ ইন আ পার্টিকুলার subject is biology or subject is to understand human biology or plant biology. The more you go up in biological status, the more you will figure out they are all basically coming down to same. Evolution has preserved in that way. So it's always same. So আমার একটা জিনিস আমি বলতে চাই যারা রিসার্চ করতে চায় তাদের জন্য from a research perspective research কিন্তু is not for everyone. So let me first clear off that. Research is a strenuous job. If you want to do internationally acclaimed, known research, it's a strenuous job. And respect each scientist for that, or each person who is doing that for that. আজকের ডেটে COVID-19 এর সময় আমরা অনেকটা জানতে পেরেছি যে সায়েন্টিস্টরা অ্যাকচুয়ালি কি করেন এবং প্রবাবলি সায়েন্টিস্টরা কি করতে চান কিন্তু মনেটা হচ্ছে সায়েন্টিস্টদের জীবন COVID-19 এর আগে যেরকম ছিল এখনো সেরকমই আছে আমরা আগেও যা করতাম আমি এখনো তাই করি কারণটা হচ্ছে COVID-19 এসছে COVID-19 চলে যাবে একটা সময় চলে যাবে বা হয়তো থেকেও যাবে যেমন বাকি ভাইরাল ইস্যুগুলো রয়েছে তাতে ভয় পাওয়ার কিছু নেই ব্যাপারটা হচ্ছে আমরা এর জন্য কতটা প্রিপেয়ার আর সায়েন্টিস্টরা সেই প্রিপারেশনটা দেয় আমাদের কিন্তু সায়েন্টিস্টের লাইফ নর্মাল নয় আমি আগে থেকে বলে দিচ্ছি একটা সায়েন্টিস্ট যখন সে PhD করুক বা সে সায়েন্স ল্যাবই চালাক সে কিন্তু দিনে অলমোস্ট 24 ঘন্টার মধ্যে অ্যাটলিস্ট 12 থেকে 15 ঘন্টা অ্যাক্টিভ থাকে মেন্টালি অ্যাক্টিভ থাকে এবং সেই মেন্টালি অ্যাক্টিভিটির মানে হচ্ছে নট অনলি অন বেঞ্চ অফ বেঞ্চ আমরা পড়ি আমরা পড়াই আমরা রিসার্চ করি সো ইটস অল রাউন্ড অ্যাক্টিভিটি সো 12 ঘন্টা থেকে 15 ঘন্টা ডেডিকেশন একমাত্র আসতে পারে if you really wanted to do science. If you don't, you will automatically be deleted from the system. Because সায়েন্স পড়তে গেলে সায়েন্সের প্রতি ভালোবাসা থাকতে হয়, কারণ ভালোবাসা থাকলে পরে আরো জানার ইচ্ছে হয়, আর জানার ইচ্ছে হওয়ার পরে তখন তুমি এক্সপেরিমেন্টগুলো করতে চাও। কিন্তু তুমি যদি সেই জানার ইচ্ছেটা লুজ করে দাও তাহলে সায়েন্স করে আর কোনো লাভ হয় না, তখন ওটা চাকরির মতো হয়ে যায়। And if you really want to do job, there are a lot of jobs. But if you really want to do science, you have to know that you must dedicate yourself rest of your life for doing that. It's a dedication. But you can still enjoy family life. There's no problem with it. But that 15 hours, 12 to 15 hours of that dedication you need, and you have to be prepared mentally. So, welcome to the world of science product. And আমি বলবো যে রিসার্চ করতে গেলে কখনো আটকে যাও, যে আমার ডিভিশন সেকেন্ড ডিভিশন এসছে বলে আমি আটকে গেছি, বা আমি কেমিস্ট্রিতে ভালো নয় বলে আমি আটকে গেছি। ট্রাই টু ইভলভ, জাস্ট লাইক ক্যান্সার। এভরি টাইম ইউ ইভলভ, ইউ আন্ডারস্ট্যান্ড, ইউ নো, ইউ মেক ইউরসেলফ স্ট্রঙ্গার। যখন তুমি স্ট্রং ফিল করবে তখন তুমি আরো উপরে উঠবে। দ্যাট ইজ আ সিম্পল বায়োলজি। সো if whoever wants to do research, welcome to the world of research. And don't get limited by what you have. Use those resources to make something which is much more beautiful than what you have. Okay, that's all. Thank you.
Thank you, Anirban, for this, and especially driving home the point that research is much more than just AC ঘরে বসে কম্পিউটারে কাজ করা। It's sheer hard work, and you have to be very tenacious to have a good research career. So wishing you all the best for the future, more papers, mostly, and the fact that you enjoy it, which I ask all my friends that ask yourself whether you'll be happy doing it. Your face
Yes.
shows, Dr. Kaur, that you're happy doing it. So you have chased your passion and you're doing what you really like. And so that is what really matters. So with that, we come to the end of this webinar. Thank you everybody for staying with us for these more than 2 hours. So wish you good night and stay safe.
Good night.